Today, we are diving deep into one of the most incredible processes in the universe, DNA replication. Seriously, this isn't some dusty topic from a biology textbook. This is the story of you. It's the microscopic, high-speed, high-stakes process that is literally building and rebuilding you every single second of every single day.
So let's just start with a question that sounds completely impossible. How does a single one of your cells, a tiny, tiny thing, take 2 full meters of genetic code, your DNA, and copy it into a space smaller than a grain of dust nearly perfectly every single time a cell divides? How does it pull that off? And when I say nearly perfectly, I mean it's almost beyond belief. The error rate for this process is less than 1 mistake in a billion letters of code copied. To put that into perspective, that's like someone typing out a whole shelf of encyclopedias and then doing it again and making only 1 or 2 typos in the entire library. It's just astounding. All right, are you ready? Let's dive into this. We're going to unpack this molecular mystery piece by piece and see exactly how life's most fundamental copy machine actually works.
So first up, let's really wrap our heads around the scale of the challenge here. We'll call this section the copying conundrum. The thing that's so staggering is that it's not just about accuracy, it's also about speed and incredible packaging. I mean, a bacterium like E. coli can copy its entire genome in about 20 minutes. Your cells have to copy a genome that's almost 1,000 times larger, just as fast, all while it's crammed inside the tiny nuclei. So, to solve a problem this huge, life has evolved this elegant team of molecular machines. Think of it like a highly specialized construction crew, where every single enzyme has a critical job to do. So, let's meet the team. First on the scene is helicase, and the name says it all. You can think of this enzyme as the unzipper. It latches onto that DNA double helix and just motors down the middle, breaking apart the bonds that hold the 2 strands together. This creates what's called a replication fork, the little bubble where all the action is about to happen. Now, if you've ever tried to quickly unwind a twisted rope or an old phone cord, you know that as you pull it apart, the part ahead of you gets wound tighter and tighter. The same thing happens with DNA. That's where topoisomerase comes in. It's the strain reliever. It zips ahead of the helicase, making these tiny, temporary cuts to release the tension so the whole molecule doesn't get knotted up into a tangled mess. Next up, we have primase, and this little enzyme points out a really key limitation in the whole process. See, the main builder, the star of our show, can't just start copying on a bare strand of DNA. It needs a starting block. And primase provides this by laying down a short little piece of RNA, called a primer. This gives our master builder a place to land and get to work. And that brings us to the real star of the show, DNA polymerase. This is the master builder. It slides in, reads the template DNA, grabs the right building blocks floating around in the cell, and builds the brand new matching strand. And it does this with that incredible speed and accuracy we talked about. So we've got our team assembled. It sounds pretty straightforward, right? Unzip, lay a primer, copy. Done. Well, not quite. This is where the story gets a brilliant, brilliant twist. Let's look at what I call the backwards strand puzzle. Okay, here's the central problem. DNA strands are antiparallel. They run in opposite directions, like a two-way street. But our master builder, DNA polymerase, is a one-way machine. It can only add new bases in one direction. So this works great for one strand. But what about the other strand, the one pointing the opposite way? How does it copy both at the same time? The solution is it's so clever. One strand, which we call the leading strand, is no problem at all. It gets made in one long, continuous piece. The polymerase just follows right behind the unzipper. But the other strand, the lagging strand, has to be built backwards, away from the direction the fork is opening. So the cell has to synthesize it in a bunch of short, little, discontinuous pieces. This whole backwards construction happens in 4 key steps. First, primase comes in and lays down a whole bunch of RNA primers along the strand. Second, DNA polymerase jumps from primer to primer, filling in the gaps with short stretches of DNA. These are called Okazaki fragments. Third, another type of polymerase comes through and swaps out all those RNA primers for DNA. And finally, an enzyme called DNA ligase acts like molecular glue, sealing up all the little nicks between the fragments to create one continuous finished strand. It's a beautiful solution to a really tricky problem. So the backwards puzzle is solved. But there's one last complication.
It's a unique problem that really only affects cells like ours, cells with long, linear chromosomes. It's the problem of reaching the end of the line.
So just picture it. The whole replication machine is racing towards the very tip of the chromosome. The leading strand, the easy one, no problem. It gets copied all the way to the end. But what about that lagging strand, the one that's being built in little pieces, backwards? Here's the issue. When that very last RNA primer on the lagging strand gets removed, it leaves a tiny gap at the very tip. Now, DNA polymerase needs a specific chemical hook to grab onto to fill that gap. But at the very, very end of the chromosome, there is no hook. The result? Every single time the cell divides, a tiny piece of that chromosome is lost forever. It just gets a little bit shorter. Now, wait a minute. If our chromosomes get shorter every single time they replicate, why haven't we just disappeared after billions of years of evolution? This brings us to our final section, a truly fascinating story of protective caps, cellular clocks, and their surprising connection to cancer. The solution is a beautiful structure called a telomere. And the absolute best way to think about a telomere is like the little plastic tip on your shoelaces. They aren't the important part of the shoelace, but they protect the important part from fraying and falling apart. Telomeres are just long, repetitive sequences of junk DNA that act as protective caps on the ends of our chromosomes. But even those caps wear down. So how are they maintained? Well, enter an incredible enzyme called telomerase. You can honestly think of it as a molecular fountain of youth. It carries its own little instruction manual, and it adds those repetitive telomere sequences back onto the ends of the chromosomes, rebuilding the protective cap and fighting back against that shortening process. And this is where it all comes together, because telomerase activity is not the same in all our cells. In most of your body cells, it's actually turned off, or at least very low, so their telomeres shorten over time, which limits their lifespan. But in your stem cells, the ones that have to divide over and over, telomerase is highly active. And here's the dark side. About 85% of cancer cells figure out a way to switch telomerase back on, giving them a kind of cellular immortality that lets them divide out of control. So what this really means is that the end-of-the-line problem isn't a flaw in the system. It's a feature. The shortening of telomeres is a built-in cellular clock, and it's directly linked to the aging process. And figuring out how to bypass that clock, well, that's a hallmark of cancer. This delicate balance between aging and immortality is one of the most profound trade-offs in all of biology. And that leaves us with one final, fascinating question to think about. If the length of our telomeres is so closely tied to this cellular clock, what could understanding, and maybe one day controlling this process, mean for the future of human health, the future of aging, and our fight against diseases like cancer? The story of you, it turns out, is still being written.